Now look at this.
What's up, people? Penguins to do topic 6.4 on translation. So back to the central dogma. Translation is the second part. mRNA to our polypeptide is translation. So we're going to read the mRNA to synthesize a polypeptide strain. Process of translation is going to take place in the ribosomes, which will be located in the cytosol of both prokaryotes and eukaryotes. Due to the fact that eukaryotes have membrane-bound organelles, specifically the rough ER, we will find that some of the ribosomes could receive a signal that will cause them to move to the rough ER to finish translation at the rough ER. So there's three different steps we have in translation: initiation, elongation, termination. So for initiation, we're going to see that the small subunit is going to bind to the mRNA. It's going to keep on moving, starting at the 5' end, and it's going to move through looking for the start codon, AUG. Once it finds the AUG, the large subunit is going to come in and bind, as well as the tRNA with the anticodon UAC and the amino acid methionine. The large subunit of the ribosome has three sites to it. It has an A site, P site, and an E site. A site is where we're going to add the next amino acid. The P site is where we're going to hold the growing polypeptide. And the E site is where the empty tRNA is going to exit. As we move from initiation to elongation, we're going to see that the tRNA holding that methionine is sitting at the P site. The next amino acid is going to come in on a tRNA that's going to base pair up with the codon. At the A site, we're going to see that we're going to create a new peptide bond between that polypeptide that's at the P site with the new amino acid at the A site. After that happens, we go through translocation, and the whole thing is going to shift over. So what was at the P site is now at the E site to leave. What was at the A site is now at the P site, and now the A site is open, ready to take in the next amino acid. Termination takes place when you reach a stop codon. So UAA, UAG, or of course, the unstoppable UGA. Go Bulldogs. So in the AP exam, you may be asked to translate something. So as you see, this is what the codon chart's going to look like on the AP exam. Specifically, I want to notice right here at alanine. So that third codon is going to be variable. It can be any of the 4 nucleotides and still code for the exact same amino acid. That's what we call a wobble. So notice that you can have multiple codons coding for the same amino acid, but you can't have one codon coding for multiple amino acids. So here you see an example of a retrovirus, HIV. When it infects its host cell, it's going to insert its viral RNA that then uses the enzyme reverse transcriptase to take the RNA as a template and synthesize a DNA strand from it. That DNA is inserted into the genome of the host. So every time the host makes copies of its cells, specifically your white blood cells, you'll make copies of that HIV. Hence why it's a human immunodeficiency virus. Remember, I'm doing this for every single unit, so AP Bio Penguins just says